Welcome to Module 9. In Module 9, we're going to start getting into the real nitty-gritty of microbiology. Module 9 is about pathogenicity, which is about how microbes cause disease, and epidemiology, which is learning about disease with the idea of preventing global outbreaks. Much of the terminology that you're going to hear in both sections, pathogenicity and epidemiology, are going to sound very familiar to you, right? And so hopefully we can apply some of these things we learn to real world context. Okay, so with that said, let's buckle up and get started. So we're looking at an epidemic curve here. These would be COVID-19 cases and deaths in the US, I guess, from March 1st to October 27th, right? So, one of the things that I want us to get an appreciation for is how do we talk about some of the statistics, again, on a surface level, because epidemiology is like this really deep field, all about numbers, right? So sometimes you'll see data thrown at you, and then if you're not really paying attention to how the data is presented, it can really be skewed to mean anything that you want, right? There's this old saying where we say there are lies, damned lies, and then there are statistics, right? Because you can find a statistic and really make it seem any way that you want, right? It's common to often throw multiple pieces of data on the same graph with this, orange line being the daily cases, and then this grayish, blackish line being number of deaths. And if you're not paying attention, you're like, holy, look at the number of deaths. For some reason, it's way higher than the number of cases, right? But our scales are different, right? So here we have 70,000, and then here we have 2,000, right? So that's definitely something to be aware of, right? It's presented in such a way so then for you to be able to see the peaks and valleys of deaths relative to cases, right? Because if we were to graph the number of deaths, it would be this very low line down here somewhere, and you really wouldn't be able to see the relative deaths to the number of cases. The other thing, and that's a very common thing, probably a better way to do this, is this idea of a 7-day average. And sometimes we don't understand that either, right? If you take daily average or daily number of cases, it would be up and down, and then it's really hard to get a trend because, you could have, let's say, a low point because there are less testing done that day, or maybe there are indeed less positive cases, right? But a 7-day average takes the average of the last 7 days and puts that into a plot here, and then let's pick actual values, and this day here will be like the 7 days before that, and then this day here will be the 7 days before that, right? And so then you can get a really neat look at then not just the one single day, but how did the last 7 days look, and then that presents a much better depiction of the trends, right? And so if we look at our data, how does one erase this? There we go. So then we can start to really look at trends, right? So we're looking at the number of cases. This was our very first peak of COVID-19, and then we hit our shutdown sometime around here. Notice that decrease, and then after we reopened sometime in June, we're seeing that second, right? And then people started to freak, and then we started to shut down, and then now we're in this current upward trend again, right? So the 7-day averages help us much better visualize the trends, or the moving average is what we often call it. Okay, so that's kind of this idea of epidemiology. Again, it's a large field. Let's jump into pathogenicity. Let's talk about disease and then how microbes cause disease. The term disease means a condition that's basically you're impaired or you're not normal, right? And really it's disease can be sometimes quite subjective, as we'll talk about in a second, right? There are many causes to disease, right? So, there's infection, of course, microbial disease. There are genetic causes to disease. There are environmental causes, et cetera, et cetera, et cetera. So many things that can impair you that sometimes it's really hard to find out the actual cause, right? And many things kind of seem very similar. We, of course, are concerned with microbial disease, right? So we care about infection. And basically a disease because a microbe has been able to colonize you, for the most part, right? Which means they live, they grow, they replicate. However, whatever term you want to use, it's all more or less the same, whether it's for a long time or very short time, right? But the pathogen persists. There are some kind of variations of microbial disease. You can get microbial disease without colonization. So something like tetanus, for example, or botulism, it's the toxin or the product of the microbe that causes disease, right? So if you ingest some botulinum toxin, you can get botulism even though the bacteria may not be around, so it's not really an infection, right? It's what we call an intoxication, but we still do call it a microbial disease. So once again, elements, you have to always understand the situations that you're in. When you have disease, of course, there are things that happen to you, right? And we often call these things signs and symptoms. Typically, the word here we use synonymously to mean both are symptoms. They always say, hey, what are your symptoms? What are you feeling, right? But the correct terms are signs and symptoms, and even though they sound the same, they're actually slightly different, okay? So signs are measurable, right? They're objective, and they can be observed, right? So anything that can be measured, whether it's something that's seen, that's measured as well, right? Observe. Temperature, heart rate, blood pressure, a rash, a cut, anything like that, although a cut technically isn't microbial in origin, right? But it can often lead to diagnosis because of certain, we'll call it transmission, right? But anything that can be seen, observed, or measured is a sign, right? So there's, one can say that, and then on the flip side, symptoms are characteristics that are felt only by the patient, right? So they're subjective, right? They cannot be measured. And so some very common things are things like pain and nausea, appetite, right? There's no measure. They can't go and take like this thing and they poke you and then they say, okay, you are at pain level 10. I can feel it based on my pain meter, right? It doesn't quite work like that. And so, you think about every time you go see a doctor, what do? They start asking you, how you feeling? How you been sleeping? You eating much, right? All that kind of stuff. And does it hurt to walk? Right? And so what they're doing is they're assessing your symptoms, right? How do you feel? At the same time, they'll start picking up and rotating and looking at stuff, right? And then once you say something, say, oh, maybe let's go measure the blood pressure real quickly. Let's go measure the heart rate, check all these things out, right? So then they can have this combination of signs and symptoms. So some of the kind of the difficulty of diagnosing diseases is that sometimes, especially the symptoms, can be subjective, like very subjective, right? And we all know that person who is like in perpetual pain or always sick or always not feeling well, right? Whereas, you might be that person, like when you get a cold or flu, you're like running around still doing your yard work because you quote unquote feel fine, right? So, there's all these things. They do try to quantify stuff like pain, right? So this Wong-Baker pain scale, if you've ever been to the doctor, we've all been to the doctor, but sometimes they'll like give you this sheet and it says, which one describes you on this thing, right? Then you're like, oh, I don't know. I'm definitely not this one, but maybe this one. So let's call it a 6. And they, based on that, then they'll try to assess kind of the level, right? And then again, some of the diseases that cause very mild symptoms, right, can sometimes be pass unnoticed because, for someone who might be here, others might be here or even there. They feel totally fine. So, and that's always some of the challenging aspects of diagnosing diseases. A syndrome is a group of signs and symptoms associated with a particular disease, right? So it's oftentimes like, oh, you have fever, you have chills, you have this, you have that, you have pains, blah, 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 right? All those things kind of come together. And then so we call that a syndrome, right? Sometimes a syndrome can have many possible causes. So something like toxic shock syndrome, for example, you have these various things that are related to shock. And then the cause of the toxic shock can be quite different. And so that's kind of the challenge in trying to diagnose, right? Sometimes the syndrome themselves will just describe the disease. Typically not microbial diseases, right? But something like Down syndrome, for example, it's called Down syndrome because it's these characteristic signs and symptoms that are combined, right, that lead to a certain disease, right? But all those signs and symptoms really only point to this one disease, right? So again, sometimes the terminology between syndrome and signs and symptoms and disease can be kind of gray, just depending on the origin of where it started. Kind of what I just alluded to, you can have disease and be asymptomatic, right? Or what's called subclinical, no noticeable signs or symptoms, right? And again, now here, the key word is noticeable, right? Of course, No signs means that you have no signs, means you no fever, blood pressure's okay. All these things, at least that you thought to look at, are normal, right? But there are no symptoms, right? So, someone who, again, you could have a cold and feel fine and just going to go about your day, and then someone else can have a cold and they're bedridden and they can barely walk, right? So sometimes these things are slightly, again, very subjective, right? And so with the case of COVID-19, for example, there's definitely evidence of asymptomatic spread, right? People who feel no signs and symptoms and are able to spread the disease. But sometimes it's been also described as pre-symptomatic, right? Because, maybe this person will eventually get signs and symptoms, but it didn't really bother them very much to the point where they even felt it, right? And there's a lot of stories of people saying that, I felt totally fine, right? But then like 2 days later, all of a sudden, bang, they felt really bad, right? So again, when being asymptomatic means no noticeable signs or symptoms, right? And so when you diagnose something, it's always a combination of signs and symptoms and then exposures and where you've been in this environment. Sometimes it's very easy to pick out right away. It's like, oh, for sure, everything points to this one disease. Sometimes it's very difficult, right? Because there's so many things that cause the same signs and symptoms, right? So, if you look up, I think we've all done this, right? You got this, I got this weird pain in my back, right? And then it's like, it kind of hurts a little bit on my knee. And then you start going to WebMD and looking it up. And next thing you know, you have cancer, right? Or something like that. You're like, oh my goodness, right? What the heck, right? And because there are, again, many the layers and levels of these things, right? Which is why it's always dangerous to self-diagnose. Because, you take information and you take it the wrong way. Next thing you know, you're writing your own will and freaking out and calling everybody, telling you love them too much, right? So, it's very important then that everything gets put together, yeah? I just want to show this. We don't have to memorize this, right? And in fact, many of you are going to go on to take things like medical terminology classes, right? But the things that are said, they mean something, right? So when you have something like hepatitis, for example, it's not some sort of strange word that they made up, right? The roots, prefixes, and suffixes all mean something, right? So this word itis, right? That means an inflammation, right? So if you have like brainyitis, right? Sometimes we say, oh, I got brainyitis. Yeah, it just sort of sounds smart, right? Which then means inflammation of the brain, right? Fingeritis, right? Anything itis means some sort of inflammation, right? See the suffix omas are always some sort of tumor, right? Derma's got something to do with skin, right? Cyto meaning cell, hepa meaning livers, right? So all these things kind of get put together, and then you put them together, and then you make whatever the characteristic of the disease is, right? And so, as you move on in your career and as you start to learn these things, when someone says, oh, this person has neuropathy, right? Then you're like, oh, hey, I know what that means, right? It's a disease of the neurons, right? So, you kind of learn these things. So again, there's kind of this systematic way of talking about things, okay? So with that said, let's define a bunch of things. So much of what we're going to talk about are going to be definitions, right? So let's define some things. Let's talk about how we classify disease that microbes cause. We are talking about infectious disease, right? Which is the disease caused by pathogen. Again, many, many types of diseases. We care about the microbial cause in a microbiology class, right? Now, not all infectious diseases are the same. Some are communicable, right? Which means they can be spread from person to person. I think of it, when we communicate thoughts from people to people, right? A communicable disease means that it can be spread from person to person. Kind of related to that, but a little different, is this idea of a contagious disease, right? Contagious diseases are communicable diseases that are spread easily, right? So something like HIV or AIDS, HIV is communicable. It can be spread from person to person. Same with hepatitis, right? But the mechanism of transfer isn't that easy. You kind of have to acquire it. It's kind of this work, this effort to get it, right? Whereas something like a cold or measles, highly easily to spread, right? Measles is an excellent example of a contagious disease because it spreads very easily, right? On the flip side, a non-communicable disease is not spread from person to person. So there are many diseases, something like tetanus, for example, that if you're hanging out with someone who has tetanus, you're not going to get tetanus from them because it doesn't spread from person to person. right? And then finally, a zoonotic disease are transmitted from animals to humans, right? And sometimes it's called a zoonosis. So when we classify diseases, of course, we talk about how it can be transmitted, right? But we can also talk about where we get them from, right? And sometimes, and it seems somewhat counterintuitive, right? But sometimes we get diseases, most of the time we get it out in the community. We call these things community-acquired diseases, right? But sometimes we get it from our hospital settings, right? And the hospital setting is, really, if you think about it, this perfect storm for being able to pretty much get sick, right? A lot of sick patients are there, therefore many potential pathogens, right? If you don't properly clean equipment or the linens that are shared amongst these many sick people, right? It's this potential for spread. If the worker, the nurse or the doctor, they don't properly wash their hands, properly sanitize, they're too busy as they're running around taking care of the 30 patients that they have, right? Once again, they can now spread these potential pathogens from patient to patient. On the flip side, patients tend to be weakened or compromised, whether it's because of surgery, because they're opened up, or because of this high use of antibiotics, right? And we talked about superinfections, this idea that everything is compromised, your normal microbes are not around, and thus leaving you prone to an infection by something like Clostridium difficile. Right, and so these are what we call nosocomial infections that happen that are acquired within the hospital setting. Right, a bit more specific is an iatrogenic disease. Right, this is an I, by the way. Sometimes we think of it as an L, but remember it's a beginning of the sentence. So it's an iatrogenic disease, and these are technically a nosocomial infection, result of a medical procedure. Right, so typically you have surgery and then you get infection of the wound, something like Clostridium perfringens, for example, very common iatrogenic disease pathogen, because of the fact that, once again, it's not super invasive, right? But if you give it that opening, you cut someone up, right? All of a sudden now it's a much easier access. Clostridium perfringens, right? Or maybe some sort of catheter that was kind of inserted, but the catheter wasn't cleaned properly. That would also be an example of an iatrogenic disease. Okay, so when you get sick, illness goes through these various phases, and it's related to the load of the pathogen, right? So typically, when the pathogen level is low, you're not sick. As the pathogen starts to increase, you start to get sick, and then so on and so forth, right? So it goes, disease will progress through these 5 phases, right? Starting with incubation. And incubation is this initial entry of pathogen, right? So somehow you got exposed, and we'll talk about how diseases get transmitted and then how they have to enter, right? But this incubation period is when the pathogen comes in. It's at this point the pathogen is fighting with your immune system. And we'll talk, I believe it's the next module or the one after that, about your innate immune system and your adaptive immune system, right? But there's this battle right now, right? And the initial entry is all with your innate immune system. And if it can overcome those things, if it can overcome the barriers and the defense systems that you have, you are going to get sick, right? So at this stage, then the pathogen enters, it starts to replicate, right? So we have our increase in the pathogen load, and then you will start to feel sick. During the initial incubation period, you show no signs and symptoms. Generally, you're also not transmissible. So generally, at this stage, you don't transmit the pathogen. As the pathogen is still trying to get its foothold, it has not yet colonized you. Right? The length varies, right, according to the disease. So some pathogens that are extremely fast replicators or have these extremely invasive factors that allow them to cause disease, they will have a very short incubation period. Some pathogens have a much longer incubation period. The prodromal period is the onset or the initial signs and symptoms. As it starts to replicate, now your immune system is activated. So we go from an innate immune system to an adaptive immune response. Now the battle really begins, right? And at this stage, it's kind of, are you going to feel sort of crap, or are you going to feel incredibly crap, right? And so this is where the big fight happens between pathogen and immune system. Depending on the disease and the pathogen, you may be transmissible at this point. You may be able to pass on infection, but you may not have very severe signs and symptoms, right? And again, it's really all dependent on the disease. As the pathogen starts to replicate and continue to grow, you now go through the period of illness, right? Which, as the name implies, this is when you're sick, right? You have the most obvious and most severe signs and symptoms. This is generally now when you're highly transmissible. You know you're sick, right? And then your immune system is actively working to try to fight off this infection, right? Hopefully your immune system wins. And then we go to this period of decline, right? Where our signs and symptoms start to decline as the pathogens start to decline, right? So the pathogen starts to get worn out. It starts to lose the battle, right? And then your immune system now is starting to win and turn the tide here, right? The decline phase is typically when we're kind of over the hump and starting to feel a little bit better, right? But because your body is in this weakened state, your immune system really is only good at fighting one infection at a time. Here you're susceptible to secondary infections, and we'll talk a little bit about what secondary infections are a bit later, right? But it's the period of decline when you are prone to those types of things. And then finally we have our period of convalescence, right? That's when everything kind of returns to normal function. Once again, depending on the illness, you may have like kind of residual signs and symptoms or damage left behind that needs to be fixed. So sometimes you have these long, what we call tails of the disease, right? Think of this as a tail. So this would be normal. This is kind of where we're almost normal, and then sometimes it takes just a long time to get there. So COVID-19 for some people have these long tails. They'll report respiratory issues with difficulty breathing like months after the pathogen has left, right? And so then kind of getting to this whole infectivity is very disease dependent, right? Each stage is very different for each disease, right? And then sometimes it's even person dependent, right? Each person will experience a disease quite differently, right? So sometimes when we say, oh, stop being such a wimp, I had the flu, it was totally fine for me, right? But our genetics, our environment, everything will kind of factor in to how one displays and feels this disease. All right, so kind of related to this is how long a disease lasts, right? And so it's like how long the duration of the disease is, right? Most diseases are acute, right? They're relatively short, typically a rapid onset, typically a short incubation. These types of pathogens tend to grow very fast, right? And at the same time, we tend to clear them very fast, right? So short. The way I like to remember this is acute diseases are acute disease, short and sweet, right? On the flip side, chronic diseases occur over long periods of time, right? So sometimes you'll feel these signs and symptoms. Typically, they develop slow. So it's not like you have this rapid onset and you're sick, and then you're like sick for long periods of time. It tends to be like start slowly and then make its way worse over time, right? But you're constantly feeling these signs and symptoms over long periods, which sounds similar to a latent disease, but it's different. With latent diseases, it's a dormant period, right? So no active signs and symptoms. And then the disease can reactivate later. So HIV, perfect example of a latent disease, right? Where you'll have no active signs and symptoms. You'll actually have the short acute phase with your first viral infection, and then you'll have no signs and symptoms. And then later in life, HIV then becomes this chronic disease where you start to slowly feel the symptoms of AIDS as the HIV has kind of activated. Okay, I want to talk just a tiny little bit about Koch's postulates. We're not going to get into the details of this, so I only want to mention this just so you know what they are. Oftentimes they'll be talked about. We don't need to talk about the details, right? But Koch's postulates are kind of like these set of experimental rules that link a pathogen with the disease, right? And sometimes it seems quite obvious and self-explanatory, but it's actually something that it took some time for people to be convinced that this is how we should do it, right? So basically, if you have a diseased organism, very first thing you need to be able to do is find your suspected pathogen within a diseased organism. So you need to be able to culture it and then grow it, right? So that's one. And then every case of the disease must have this pathogen, right? So if you have a bunch of people and you think, oh, I think this virus is the cause, right? But you find a whole bunch of people that don't even have that virus, well, it's obviously not the cause, right? It has to be in every single diseased organism. and then taking that virus or that pathogen, then it can be a bacterium, it can be a virus, it can be anything, and then you stick it back into your test organism, typically a mouse or something like that. It needs to be able to cause the same disease, and then once again you need to be able to pull out that same pathogen, right? Something along those lines, right? This is a very experimental process. There are many changes to this with new molecular techniques and that kind of stuff. There are many challenges because not all microbes are culturable, right? But nowadays it's the molecular techniques that are often found, right? So typically viruses, for example, very rarely do we get the virus, right? It's often the nucleic acid sequence that leads to the initial assessment, right? So a good example of this was SARS-1, right, back in 2002, 6, I forget, somewhere around there, right? It took a long time to figure out what was causing the disease. They initially thought it was influenza, but eventually someone said, hey, maybe it's a coronavirus, right? And they're able to go and pull out the organism, do a bunch of things, and then identify it, right? It took a long time. With COVID, right, oftentimes called SARS-2, it was much faster, right? So once again, it was called an abnormal pneumonia, and they really weren't sure what to look for. But then someone suggested, hey, how about we look at coronaviruses? They did a very quick sequencing of some of the patients, and then they found the same coronavirus sequence in every single one of these, right? So this whole arc happened in like a week, as opposed to before it took months to figure out what was actually causing that disease. I may have these timelines wrong, right? But definitely much faster using molecular methods. Okay, so I talked way too much about this, right? I didn't want to, but Koch's postulates, again, the purpose of this is to link a pathogen with the disease that it causes. Okay, so we're going to move on to pathogenicity and virulence, right? We'll talk about what some of the things that microbes do to cause disease, right? Pathogenicity is all about the ability of microbes to cause disease. With that said, I have a meeting to run off to, so I'm going to take a break. You can take a break if you want to. You don't have to, right? So I'll see you I'm going to be back in like 2 hours, but maybe I'll see you in a second. All right, so let's talk a little bit about pathogenicity. I kind of forgot where I left off, but I'm back, and I'm pretty sure you didn't go anywhere. So pathogenicity is the ability of a microbe to cause disease, right? So to generate pathos for disease, right? So, and then we use this term virulence to describe the degree to which an organism is pathogenic, right? So something that's highly virulent means that it causes severe disease, right? Or causes disease readily, whereas avirulent or low virulence means it does not, right? Sometimes these terms are used interchangeably, right? So typically, but the distinction here is that if we have, let's say, an organism that is non-pathogenic, right? We would say that it cannot cause disease, right? But some strains, like the Streptococcus pneumoniae, for example, is pathogenic. It's able to cause disease. But then we have strains of the Streptococcus pneumoniae, that S strain and the R strain, that is either virulent, right? It is able to cause disease, right, to some degree, or avirulent. It can no longer cause disease to any degree, right? So the bacterium itself is pathogenic, but the different strains have a certain level of virulence, okay? And then things like mutations or inactivation of genes can lead to lower virulence, right? So again, going back to the Streptococcus pneumonia example, this is that Griffith's experiment, by the way, if you forgot what I'm referencing. These mutations often lead to, the smooth strain is virulent, right? And then the rough strain is avirulent. And that's because it has lost the ability to make capsules. So generally mutations will lead to a lowered virulence. On occasion, it can lead to increased virulence, right? But so over the years, that's how new strains can come about, and that's how different things can happen, right? New pathogens. But generally, mutations will lead to a lowered virulence, right? And so we'll talk a little bit about how mutations can lead to maybe not necessarily higher virulence, but different virulence, okay? We can quantify virulence, right? We always like quantities and like to quantify things. So we have this thing called a median infectious dose, right? Sometimes just called an infectious dose or simply ID50. And what an ID50 refers to is the number of pathogens that can cause infection in 50% of test animals, right? And this is a kind of this vague number, this kind of range of this number, because we have to understand that if in any one population, there's going to be a different susceptibility to disease. So if we say that, how much of any one pathogen does it take for all the animals to get sick? Well, some of the animals that tend to either have a stronger immune system or just for some reason be less susceptible because of various mutations in their own cell structures, right? That number might be way too high. So this practical number, this ID50 says, well, about half the population is going to get sick, right? So then that kind of leaves this variation. And usually it's test animals like a mouse or guinea pig or whatnots. When we're testing things like toxins or things that will kill, right, not necessarily cause disease, we refer to the medium lethal dose or LD50, right? And again, it's a median value, right? The actual value will always depend on many, many things, right? So if you are, age, for example, really young, really old, the ID50 might be much, much higher than is required. Health. If you have if you happen to be healthy today, whereas tomorrow you happen to be sick, and once again, the ID50 or the LD50 can vary according to that. Your immune status. We all have slightly different immune systems, right? And sometimes we think, by the way, that, I have a strong immune system, I don't get sick. Well, actually, you don't want a too strong immune system. We always call it the Goldilocks zone. You want an immune system that is just right. And we'll talk about some things that happen with immune systems that are quote unquote too strong, right? So, it's somewhat like Neanderthal like to think like, oh yeah, I'm strong, that's why I don't get sick, right? Well, actually, you know what? Pathogens really don't give a crap how much you can bench press, right? It's the immune system is very, very fine-tuned and very different, right? So, your ability to rebuff disease really is dependent on so many things that have absolutely nothing to do with physical strength, right? And so that's this common misnomer. And we'll talk about some elements of immune system and immune status in future chapters. And then route of entry is very important. Some pathogens are highly virulent, right, through some routes of entry and maybe less so through others. And we'll talk about that in just a second, right? However, the ID50 doesn't necessarily correlate with disease severity, which we'll talk about right now, right? So just some examples of ID50 values, right? Don't memorize this chart, okay? Please don't, right? We're just going to use this as like an example, right? So, but we want to use some very quick examples. So here we have, for example, the one that I want to talk about. So here we have Salmonella enterica, Typhimurium cerovar, and then here we have the Typhi cerovar, right? So two different cerovars of Salmonella, right? Cerovars are basically, they're pretty much the same, but they have slight differences in their expression on the outside, right? So, Salmonella enterica and Salmonella enterica, same species, slight variations, okay? So, between the two cerovars, Typhi and Typhimurium, which of these two cerovars is more virulent? Which one can cause disease easier? If you said Typhimurium, you would be correct, right? So let's think about what this number means. And we'll ignore, again, I don't like ranges and stuff. We'll keep it simple, right? So 1 versus 1,000, right? So this means that one single cell of this Typhimurium strain or cerovar can cause disease in about 50% of the population, whereas it takes 1,000 of the Typhi cerovar to cause disease in 50%, right? So it takes a lot more of the Typhi cerovar to cause disease, right? So thus, the Typhimurium strain is more virulent, right? However, the Typhi strain or cerovar actually causes disease that is much more severe, right? So again, the ID50 doesn't necessarily mean that it causes more severe disease. It just causes disease easier or better or whatever you want. It takes less of them to cause disease, okay? So lower the number means more virulent. On a related note then, we can look at LD50s, right? So here we have LD50s of a number of toxins, right? And LD50s are always referenced with the weight of our test animal, right? So whether it's weight of human, weight of mouse in this case, because the weight, of course, also is important, right? Because bigger things are going to take more toxins to knock it out, right? So you really, if I were to ask which of these three is the most toxic, right? You would almost immediately eliminate this one because that's in the middle. So it's either going to be the highest number or the lowest number, right? So just like with ID50s, the lowest number is the most toxic, right? So 0.03 nanograms is all that it takes relative to 1,350 nanograms, right? Let's go fun fact, right? Botulinum toxin actually is the most potent biologically produced toxin known to humans. The most potent, right? This teeny tiny drop of botulinum toxin is enough to kill like a room of 1,000 people, right? If you were able to split it into 1,000 and then give it to everyone. So it's incredibly toxic, right? And I think we'll talk about it later, but it causes this paralysis, and then pretty much eventually gets to this point where you can't even breathe because you can't move your lungs, right? Totally messed up. Botulinum toxin is more famously known as Botox. We've all heard of this, right? So, if you're one of those people that hate those ugly wrinkles, right? This is what happens, right? And so actually botulinum toxin was initially, and it is still used, by the way, as a therapeutic for things like migraines, which, at some part someone realized that migraines are caused for some people because you have these intense muscle spasms in your head, right? So you have this incredible spasming of your muscles in your head, and it causes these intense headaches that we call migraines, right? And so for those people, you can take a little bit of the Botox, stick it in there, and remember, it causes this paralysis, right? So it causes these muscles to like relax and then help relieve the migraines caused by this intense muscle spasm. Right? Along the way, someone realized, hey, you know what? Every time we stick it in there to relax the muscle spasms in their head, you know what also happens? They get so relaxed that these disgusting wrinkles that make people look horrible, they go away, right? And then the genius really said that, hey, we can make so much more money marketing this thing as a cosmetic than we can as a therapeutic, right? And so that's where this whole Botox thing has exploded, right? So if you are the kind of person that injects Botox in your face because you hate these disgusting, ugly wrinkles, right? Understand, botulinum toxin, the most potent biologically produced toxin known to humans, right? And it is possible to overdose and die from this. Just leaving that out there, folks. So in the elements of disease then, right, as pathogens are coming in and invading, right, we have various levels of pathogen, right? So we have our primary pathogen. So this primary pathogen is a good pathogen. It's going to cause disease regardless of host defenses, provided, of course, that it is above, it meets whatever number is required, right? So, it's got our ID50 plays into it, right? The immune system plays into it, right? But provided we have enough of this pathogen that can then overcome the immune system, the primary pathogen will cause disease. And it'll do so using these various factors that allow it to fight the immune system, which we will talk about very, very soon, right? Opportunistic pathogens, on the other hand, they can only cause disease in what we call compromised or weakened hosts, right? Hosts that with a normal, strong immune system, right, a normal, capable immune system, would normally fight off an opportunistic pathogen, right, to the point where you don't even feel it. But if you are compromised or weakened, for example, age, right, that can be a compromise, some sort of immune deficiency, and we'll talk about some of those later. A previous or current infection. Remember, the immune system fights only one infection at a time. So if you have a current infection or a previous infection that now predisposes you, or an open wound, and we'll talk later about how like our skin is an amazing defense or barrier, right, or location. Right, so some of these things will all kind of play into allowing this opportunistic pathogen, normally weak, normally unable to cause disease, can now cause disease, right? The thing that's really bugger is that some normal microbiota can cause diseases or opportunistic infections, right? When they move to the wrong place. So good example is E. coli. Oh, E. coli always gets a bum rap, but E. coli is our friend. E. coli lives in our guts. It helps us digest things, right? So it makes vitamin K for us, right? So E. coli, we all have him, right? Vitally important for us. In fact, if you remove E. coli from our intestines, we don't do so well, right? Because we need E. coli. However, you take E. coli and move it to your urinary tract, right? Shouldn't be there. All of a sudden now, E. coli goes from being a normal microbiota, right? Something that is totally harmless, right? Commensal bacterium, commensal bacterium, then to an opportunistic pathogen, right? Causing urinary tract infections. So, and there are many examples of something like Staph aureus or Staph epidermidis, which is fine on your skin, but get it deep into your tissue, all of a sudden causes disease, right? So many things factor into what an opportunistic pathogen, the type of disease that it causes, and what opportunity is required. Okay, so let's talk about pathogenesis then. What are the stages of pathogenesis, right? And so generally, we want to think about the many mechanisms or the things that a microbe needs to do to get there, right? One, exposure. It has to get into where it needs to get to. Then it needs to be able to stick. That's what adhesion refers to. Then it needs to invade, right? And once it invades, because it needs to get to where it needs to be. Sometimes it's the place where it sticks. Sometimes it needs to go a little farther. And then finally, infection. And infection is when it's actively replicating and growing, and that's when you see disease. Okay, so we're going to go through these 4 stages as we talk about disease. Right? Talk about some various elements. Such a cringy picture in your book, right? But I fix it up for you, nice and good, right? So exposure is essentially contact with the pathogen, just like what is implied, right? Importantly, the pathogen needs to be able to come into the body, right? So this is what we call the portal of entry, right? And a portal of entry is the area or opening that the pathogen has to pass into to get into the host tissue, right? And so there are many areas. The most important of these is the mucosal surface, right? So, you think about really humans are like this one long tube, right? We've often described this tube from mouth to anus, right? So that's kind of what a human is, right? And it's all those things are lined with mucosal surfaces. We'll talk about the role that mucosal surfaces play in protection, right? But, if it can get entry through the mouth or somehow in your intestinal tract, right? Access to those mucosal surfaces are very good for many pathogens to invade. We also have the mucosal surfaces of our lungs, right? Our respiratory tract, trachea down to our lungs, right? So those are also very good. And then our conjunctiva, right? Our eyes. Those are also technically mucosal surfaces, right? So those are all areas that our pathogen get into, right? Keep this in mind though, right? Just because, something enters through the respiratory tract, most of the time it's us who puts it in there, right? So when we pick up our pathogens and then we put it into our body, it's really us who introduces it to this area. Another very common route is, the parenteral route, however you want to pronounce this. And these are breaks in the skin, right? So something like a needle stick, for example, or a cut, right? All of a sudden now we have access past our skin, which is such a lovely barrier, right? These would then allow microbes to come in, right? And then, for some it's the skin itself, right? We don't often talk about the skin actually being a portal of entry, right? But especially some fungal pathogens that infect the surface of the skin, things that cause dandruff or ringworm or things like that, they just need to be on your skin and they'll just grow right there on your skin, right? So in many ways, they don't really have a true portal of entry per se, but that is kind of their way to enter. All right, once the pathogen has entered, right, now it needs to stick. And adhesion is the ability to attach to a surface, and he uses special factors called adhesins, right? And adhesins are just usually some sort of protein, some sort of carbohydrate, like a glycoprotein, glycolipid, glyco referring to carbohydrate. So many examples of these are things like fimbriae, flagella, cilia, capsids, basically anything on the outside of the cell, right? Not things like membranes, right? Remember, membranes are inside the cell, right? Under the cell wall, right? But anything like the cell wall, for viruses like the capsid, those structures that are on the outside, all those things can act as adhesins. And what they do is they bind to receptors on host cells, right? And the receptors, so think about it now, the host is receiving, right? So the receptors are on the host cell, and the adhesion are on the pathogen. They use that to stick to receptors. Some microbes will use things like biofilms, right? So we started one of our chapters talking about the Streptococcus that forms plaque on your teeth. That plaque, that's a biofilm, right? It helps that bacterium stick to your teeth, right? Prevents that washing, right? So biofilms can act as adhesion factors. And if you forgot what a biofilm is, you can go back to chapter 9.1 or section 9.1, and it's all in there, right? But the sticky surface helps it stick. All right, once it sticks, now our pathogen needs to invade, right? So there's this spread as a pathogen into host tissue, right? To allow it to colonize. Sometimes the bacterium will colonize where it sticks. Sometimes it needs to invade farther in, right? And from that, to do that, it'll use a number of factors like toxins, enzymes, various things. Here's a really interesting pathogen, Helicobacter pylori, causes ulcers, right? And what it does is it lives in the stomach, right? That's where we get our ulcers. And we have like this really acidic mucin gel-like layer that kind of coats our stomach, of course, to help us digest, right? And this bacterium releases this enzyme called urease, which helps neutralize the pH of that stomach acid, and then allows it then to swim past this gel layer and then into the epithelial cells of your stomach, causing these holes in your stomach, right, which are our ulcers. Really neat story behind Helicobacter, by the way, right? Many thought that for years and years, ulcers were caused by stress, right? But this one person, I always forget his name, he hypothesized that ulcers were caused by a bacterium because he was able to, using Koch's postulates, isolate Helicobacter, which is really difficult to isolate, by the way. So he's able to isolate Helicobacter from many, many people that had ulcers, but not everyone. And that's why people didn't believe him, right? So in order to prove that you can indeed get ulcers from Helicobacter, I always have this weird imagination, but he drank Helicobacter, right? He probably didn't drink it, but it's called a challenge test, right? Where you challenge yourself with the microbe. So he ingested Helicobacter and he developed ulcers, right? And then he was able to isolate Helicobacter from that, right? Thus providing the link between Helicobacter and ulcers. And he ended up winning a Nobel Prize for this, I'm pretty sure. Sometimes I mix up stories, right? It happens at my age, but a really cool story, a really neat look at some of like the crazy science, which could never be done nowadays, by the way. All right, once it invades, then we have infection, right? And infection, the pathogen multiplies, right? When the pathogen multiplies, we now have infection. This is now where you're in that serious stage of having to fight off the pathogen, right? Local infections are infections that confine to a small area, and it can be like literally small, right? Like a boil or something, or it can be like small but large, like the skin, for example. If you have like rashes all over your skin, it can actually kind of be a large area, but we would still call this a local infection because it's just the skin. It just kind of spread along the skin, right? Focal infections are local infections that spread to another local site. So sometimes things like very commonly the dental infections, where you get like a prick and you get an infection inside your mouth, and then that'll spread to like another part, maybe under the skin somewhere, right? These are what we call focal infections, just a series of local infections. Often seems like systemic infections, right? But a systemic infection is infection throughout the whole body, right? I always like to think of it system-wide. So a systemic infection affects the whole body. Typically what you're going to get are things like fever, chills, shock, some kind of stuff that like affects the whole body, not just like one small area. So pain in one area is not systemic infection, right? Whereas, like a fever kind of encompasses the whole body, okay? Related to these then are primary and secondary infections, right? So primary infection is the initial infection. So your local infection would be a primary infection, right? Your focal infection, that could be a primary infection. A systemic infection could be caused by a primary infection. That's your very first thing, right? Your pathogen, your primary pathogen that causes this primary infection. Secondary infections are infections that occur during the first infection. So this is important. Happens during the first infection, and often it's due to the primary infection or treatment of, right? So we talked about using antibiotics and then getting an infection because of that, like the C. diff infection, right? We talked about opportunistic pathogens who are able to invade. So a good example of this is getting the flu, your primary infection, right? And then oftentimes now it can lead to things like pneumonia, which is a secondary infection due to your first infection. And that is much worse because your body, remember, only one infection at a time. So it's already fighting this flu. And then here comes this bacterial pneumonia, and then that tends to be so much worse, right? So flu, generally okay, right? Followed by pneumonia, it's life-threatening, right? And so actually, we call these things comorbidities. Many of the people that die from the flu in their flu season really died due to complications from the flu because that made them prone to things like pneumonia or any other respiratory infection, which then, again, much worse, and that's often the case of death. All right, so it's this stage now where our immune system is fighting our pathogen. It's trying to knock it out, trying to kill it, get rid of it, right? At some point, the pathogen needs to leave, right? So we'll talk later about how things are transmitted, right? But the portal of exit is the opposite of the portal of entry, right? So the portal of exit then is the route in which the pathogen leaves the body. And it's going to be specific to, for each pathogen, just like the portal of entry. And it's going to be related to infected tissue. Okay, so sometimes it's pretty straightforward. You're going to breathe it in, infect your respiratory tract, right? And then it's going to leave through the respiratory tract, right? Coughing, sneezing, whatever, right? So anything that leaves through breathing and whatnots, right? Sometimes it's going to be through, a stick, and then it's going to go to another tissue, and then it's that tissue then that's going to lead to its transmission, right? Most common portals of exit are contact through the skin, right? Whether it's some sort of skin infection, mucous membranes, right? So any type of GI or urogenital or that kind of stuff, those are the types of things that then are going to, leave through that same tissue that it's infecting. All right, so we've talked about, pathogenicity. We've talked about just kind of how disease progresses. Let's talk a little bit about how microbes cause disease, right? Or what factors it can use for virulence, right? And so virulence factors, in its simplicity, are simply factors that aid in causing disease. And then the types of virulence factors will determine how severe disease is, right? Because we can have many related strains of a pathogen, and they can carry different virulence factors. So E. coli is a good example of this. Pathogenic E. coli has many, many variations, all of which carry different virulence factors. So some are much more severe than others. Okay, we're going to talk about 5 general classes of virulence factors, or at least 5 general outcomes: adhesins, toxins, enzymes, evasion, and survival. And we already talked about adhesins, right? What do adhesins do? Super quick, what do they do? Yeah, right? It's for adherence, right? Adhesion is for sticking around. Usually some sort of protein or glycoprotein. Don't want to talk too much about this, right? But all types of pathogens can have adhesins. By the way, not all pathogens have adhesins, but all of them can have adhesins. And the types of adhesins that they'll have are incredibly diverse. I do not want you to memorize this, right? But again, just think of this, right? Everything has to be outside of cell, right? Anything that is inside the cell cannot be an adhesin, right? So if we have our cell, it needs to be able to interact with the outside environment. So membranes do not, right? But pili, fimbriae, some sort of protein that sticks outside, something like that, right? Those would act as adhesins to give it the ability of adhesion. Okay, invasion, factors that lead to invasion can be, once again, many, many different ways. We don't talk too much about this, right? Again, I'm not into, at least at this level, talking about some of the mechanisms, just some of the terminology, right? So the one word I think I want us to understand, and it's not a word, it's one suffix, is emia. And emia represents presence of pathogens or products in the bloodstream. Right? And in hitching a ride on the bloodstream is used then to spread throughout the body, right? So without really knowing too much then, let's define some of these words. Bacteremia. What is bacteremia? Presence of bacteria in the bloodstream. Good. What is viremia? Presence of viruses in bloodstream. Toxemia? Do we have a guess for that? Presence of toxins in the bloodstream. Excellent. And then septicemia is like this extreme case now where not only are bacteria present in the bloodstream, but the bacteria are present and they're multiplying. This is incredibly dangerous. People who are undergoing septicemia are often described as being septic, right? And septic or septicemia leads to fevers, chills, pains, nausea, incredible pains, by the way, not just like, oh, I'm a little sore, right? And then the worst case is shock. And shock is this life-threatening drop in blood pressure. So the blood pressure drops so much that you cannot adequately pump blood throughout the body, right? So shock is quite dangerous, and that's always the worst thing, right? So if someone has septic shock, it's because bacteria are just going mad throughout the bloodstream and then leading to this shock, this life-threatening decrease. All right, talk enzymes. I don't want to talk too much about exoenzymes. Once again, let's define what they are. Exoenzymes are enzymes that are outside of the cell, used to invade host tissues, right? So, we talked about what enzymes do. They perform functions, right? Exoenzymes, exo meaning out. These aren't things that they metabolize on the inside, but they use them to degrade tissues on the outside. And of course, lots of different types of enzymes with lots of different functions, right? So DNases will eat DNA, right? Phospholipases will degrade phospholipids. Collagenases will degrade collagen. Hyaluronidase is the one I want to talk about, right? So we got, our epithelial, or let's call it our skin, right, is joined together by this polymer, hyaluronin, right? And the hyaluronidase that the bacterium will secrete to the outside will start to chew away at this layer, right? And then thus kind of breaking this tight gap junction, this super tight gap between the cells, and then allows the bacteria then to slip through the outside, right? And thus get it at sensitive areas of the tissue that are much more readily, that allows them to be protected from our immune system much better, okay? And we'll just kind of leave it at that. Just generally this idea that we have these enzymes on the outside, bacteria have these enzymes on the outside that can then cause extra tissue damage, allowing then invasion into tissues. We'll spend a little bit more time talking about toxins. Toxins are biological poisons, right? And then they either help pathogens invade or cause damage to tissue. When we have damage to tissue, then they can eat the nutrients that are released from this damaged tissue, or it can cause damage to things like immune system cells or the general process, and thus causing all these problems, allowing the pathogen to thrive, right? When microbes are able to produce toxins, we say that they're toxigenic, okay? And we can get disease without presence of microbe if presence of toxin are present, right? So we call those intoxications. Two types of toxins, endotoxins and exotoxins. Basically, endotoxins are part of the gram-negative bacteria. So all gram-negative bacteria have endotoxins. Exotoxins are made by many types of pathogens, mostly gram-positive, but some gram-negative, and we'll talk about that. So let's start with the endotoxins. Super quick review, right? Remember LPS, that which are found within the outer membrane of gram-negative cells, right? This is what LPS looks like. We have our O antigen, our core, and then lipid A. This is the part that interacts with the membrane, right? So the the endotoxin, is the lipid A portion. So this part right here that acts as the endotoxin. Now, typically, it's part of the cell, so it's not going to be released out there, right? But whenever cells die, remember, cells will degrade, the lipid A will get released, right? Or whenever cells divide, every time they divide, they're going to shed some cell components, right? So when they're dividing, especially rapidly, you're going to have some lipid A floating around. Now, this is an important mechanism, right? Because when cells are dividing and some lipid A comes around, typically we'll have some low concentrations. And these low concentrations can alert the immune system, right? Because lipid A is going to trigger an inflammation response. And we'll talk later about how important inflammation is for fighting infection, all right? But again, the Goldilocks zone, right? We want some inflammation, but not too much, right? So when there's high concentrations, so when cells are growing extremely to these extremely high numbers, or when they're being killed off, whether it's using antibiotics or whether there's just so many cells, they will die, right? This excessive release of lipid A will then lead to high concentrations, which then causes this crazy inflammation response. And then later we'll learn, that'll lead to things like shock, which then can lead to things like death. Okay? So endotoxins, part of the gram-negative cell wall, low concentration, released when cells die or when they divide. Low concentrations are okay, high concentrations not so much. Exotoxins are proteins, right? So endotoxins are lipids, exotoxins are proteins. Usually they're enzymes, right? And because they're enzymes, remember, enzymes can work and then they can work again, right? So low amounts can be toxic, right? So, we're just going to go extreme. Botulinum toxin, right, has a super duper low LD50, really small number, right? Whereas lipid A has a much higher LD50, right? 2 milligrams. Right, and I don't know what test animal, I don't know where I got this number from. This is clearly different from the number I showed earlier, right? So let's just pretend it's a different, oh no, that's milligrams, right? So this is actually 0.001 nanograms, so it's similar to what I showed you. Oh no, that would be micrograms. Anyways, I'm rambling now. So don't worry about it, right? But basically, exotoxins, much more toxic than endotoxins, right? 3 types of exotoxins, which we'll talk about very quickly. Intracellular targeting exotoxins, membrane disrupting exotoxins, and superantigens, okay? Except for the last one, and actually if we talk about the immune system, the last one will make a lot of sense too. They're kind of self-explanatory, right? So intracellular targeting toxins, what do you think they do? Yeah, they're going to target stuff on the inside of cells, right? Generally, they have 2 components, the A component and the B component. So many times they're actually called AB toxins, right? And the A component is our active enzyme. The B component is the binding unit, right? So the B will bind to our cell receptor, and then the A somehow, and there are many ways this happens, the A needs to get to the inside. Once it's on the inside, it'll goof around and do whatever it does, right? So, whatever role this toxin plays, right? So we can have things like enterotoxins that affect intestinal tracts, neurotoxins that affect neuron signaling, right? Whatever the case may be, it's the B component that binds. And some exotoxins, by the way, have multiple B components. And then the A component is the activity, okay? Once it's inside, stuff happens. So I just want to talk about 2 very related but different exotoxins, so botulinum toxin and tetanus toxin. Both of these are secreted by species of Clostridium. Right, so Clostridium tetani versus Clostridium botulinum. Both of these are neurotoxins, right? So it's going to affect neuron signaling, right? So here we have our neuron. So basically in a normal cell, these neurons are going to come and then it's going to hit this thing and it's going to cause the cell to, or the muscles to contract, right? What botulinum toxin does is it goes inside and then it blocks the cell from releasing these chemicals, right, our neurotransmitters. And so when we can't release these neurotransmitters anymore, all of a sudden our muscles can't contract, right? So there's paralysis, right? Tetanus toxin, related, even though the response is totally opposite, right? So in a normal mechanism, neurotransmitter gets released, right? We now have contraction, and then we have the second neurotransmitter that tells us to stop contracting, right? That's how we turn off our contraction. Tetanus toxin, rather than inhibiting this part, it inhibits the inhibitor, right? So now all of a sudden we have this continual release of neurotransmitter. We can't turn it off. So the opposite happens. We have these spasms, uncontrollable muscle contractions. And both botulism and tetanus, they are terrible ways to die, right? In botulism, like your muscles are unable to move to the point where you can't breathe, can't blink, can't do anything. Your mind is sharp, and you're just like, and kind of float off essentially into nothingness. Whereas tetanus, like everything is firing so rapidly, right, that you literally die from exhaustion. Your muscles are constantly contracting that you can't, like, use up all your energy contracting. Terrible. Membrane disrupting toxins, what do you think they do? They're going to disrupt membranes, right? So generally they're going to do it by making holes or by destroying the membranes or the lipid bilayer, right? We have hemolysins, leukocidins, they form pores, phospholipases degrade lipid bilayer. Turns out they do other things as well. Oh, sorry, no, hemolysins were called this because they thought that hemo for red blood cells. They used to break only red blood cells, leukocidin for white blood cells, leuko meaning white blood cells. Turns out that they're able to affect many, many other cells, right? But general mechanism is the same. They're going to disrupt the membrane, cells are going to leak, and then they're going to burst, right? And so either if you're able to like kill white blood cells, you can protect yourself from the immune system. Or if you have this lovely cell that's got all the nice nutrients on the inside, you can pop those things open, you can get out all the food on the inside, right? So general idea is the same. Superantigens are basically trigger this crazy immune system response, right? And later we'll learn antigens are immune system stimulators. So superantigens are super immune system stimulators, right? So they trigger this excessive non-specific stimulation of the immune system, right? So and it does this by stimulating something called a cytokine release, right? Cytokines are chemical messengers. It's what cells use to talk to each other. We'll learn more about them later, right? So it's this incredible release of cytokines, right? So oftentimes it's referred to as a cytokine storm, just like the storm of cytokines, just all over the place, and it leads to like this intense non-specific response. So typically high fevers, pains, aches, and then once again shock, right? And once we get to that shock area, then we are in that doomed factor, right? So shock is always a bad thing, never good to be shocked. Okay, so super quick chart. You can read this on your own. Let's maybe ignore heat stability, right? But basically you can look at this on your own, right? Lipids versus proteins, right? Enzymes, specific response, non-specific, blah, blah, blah. All right, so let's talk about how various factors that allow the pathogen to survive or evade the immune system. Again, thousands of different ways. I don't want to talk about them too much, right? We will probably talk a little bit more later, right? Maybe just the capsule. We've talked about a capsule before, right? We've said before that it helps protect from the immune system. This is a super cool picture, by the way. This is a lovely stain. So these are the bacteria. This is this capsule, the sugar coat that's on the outside, right? So the capsule does a couple of things. One, it lacks like this structure that allows it to, for our immune system cells to grab. So it actually has a really hard time grabbing it, right? So it prevents immune systems from adhering, right? And thus they don't get eaten. But it also makes them much bigger, right? So when it's so much bigger, all of a sudden it's harder for immune system cell to swallow. So even if somehow they can grab it and push themselves against it, they're unable to swallow it, right? So once again, protects itself from the immune system. And there are many, many mechanisms of basically being able to avoid the immune system or being able to survive. And this is constant warfare. And again, we'll learn more about this a little bit later this semester. Talk a little bit about viruses, a little bit about eukaryotic cells, not too much because they're not super exciting, right? Viruses, they have virulence factors too. We always say they're structurally simple, but they do have factors that allow them to cause disease, right? Typically not as diverse because they generally don't have as many proteins. They generally don't have as many things that they can do, right? But they exist. And one that they all have is some sort of adhesion, because all viruses need to be able to stick to its host cell, right? Influenza uses hemagglutinin, very important, right? HIV uses something called GP120, and it's always going to stick to the receptor of our host cell, right? So host cells have receptors, viruses have adhesins, adhesins stick to receptors, right? Sticking then will lead to infection. Okay, so many, many, many ones. There's only 3 here, but basically all viruses have adhesins. there are other virulence factors. I only really want to talk about one, and it's not a true virulence factor, it's a virulence mechanism, right, called antigenic variation. And mostly I want to talk about this to clear the air a little bit on some misconceptions, right? So what antigenic variation is changes to the immunogenic portion of a pathogen, right? So the immunogenic portion, this means that this is the part that our immune system recognizes. Okay, so the immune system recognizes the immunogenic portion of a pathogen, right? And so when we clear it out, when we fight off a flu, for example, or when we get vaccinated, for example, we use this immunogenic portion, right? But if you can change or vary your antigen, right, you look different, all of a sudden now our immune system won't recognize it. And we have to do this process all over again, right? Two general ways this happen is antigenic drift versus antigenic shift, right? Antigenic drift are these small changes through mutations. And we talked about mutations, right? So it's these small changes in proteins due to mutation, right? So here we have a virus A. This would be influenza because hemagglutinin and neuraminidase, H and N, are the two very important molecules on the outside. And through some mutation, as indicated here, we now have a mutated hemagglutinin, right? So typically before, when we have our virus or our immune system recognized via antibodies, this hemagglutinin, all of a sudden now, it's no longer able to recognize that because this one is different, right? So we talked about mutation, these random changes, right? So in this case, it happened to have this random change that changes hemagglutinin such that, and remember, this is important, this hemagglutinin is no longer recognized by our immune system. However, it can still do its job of binding to the host cell receptor, right? So it's not as simple as changing. Remember, most mutations are going to be detrimental, right? So these are small little changes. Most of them are going to be bad, but occasionally you can change it in such a way where you can still do what you want to do, right? But you won't be recognized by the immune system, okay? And the flu virus is a really good example of this because it's an RNA viruses, and the flu virus is just very amenable to mutations. RNA viruses just mutate at a much higher frequency than DNA viruses. Okay, so so we'll leave it at that for now, right? Antigenic shift are changes in proteins due to gene reassortment, right? So this happens not necessarily because the viruses mate, because they don't, right? But sometimes over the course of infection, remember what happens. Virus infects, right? It then has to inject its nucleic acid into our host cell, and then it gets copied, and then it leaves, right? Along the way, a lot of errors can happen. Sometimes it'll take some host DNA with it, right? And if it can take some host DNA with it, which then gets incorporated and somehow allows it to make a small change in something like, let's say, the hemagglutinin or the neuraminidase, right? Now it has shifted, right? But more likely what happens is that sometimes two viruses will infect the same cell. So we have virus A, virus B. And then during the processing, we now have a new virus created, right? That'll have the hemagglutinin from virus B, for example, and the neuraminidase from virus A, right? So now all of a sudden we have this new virus that has never really been seen before by the immune system. And so this is what causes problems. So and these things happen all the time, right? So this is what we call antigenic shift, these large reassortments. Now, both processes, the drift and the shift, they take time. So it's not like something that happens over the course of, let's call it a global pandemic, right? So, some of the misconceptions is this idea that, COVID-2 is a coronavirus. It's mutating and now it's different from what it started. Well, mutations happen all the time. And remember, most mutations are going to be detrimental. And unless the mutation has changed some of its surface proteins, right, that allow it to continue to infect without being recognized by the immune system, it's still going to be the exact same strain, right? So that's a very important distinction. Whereas this idea that we have a vaccine for the flu, which sometimes works, sometimes doesn't, because the flu mutates, is not quite understanding this process, right? The flu has mutated over like a bajillion years, right? So out there now, we have like so many different flu strains circulating, right? So that's why, and then through monitoring, through surveillance, epidemiologists are constantly looking at birds and various animals to determine what flu viruses are floating out there that is most likely going to circulate in the human population next flu season. So they kind of have to guess, right? So because you cannot yet get a vaccine that'll infect or that'll protect you from all flus, right? So it's a very interesting statistical concept, right? Where we're trying to protect ourselves from the flu, right, by sort of guessing. And that's why we have sometimes the flu vaccine not working. Not because the virus is changing before our eyes. It's because there are so many viruses out there, you cannot possibly protect yourself against all of them. So you protect yourself against the ones that are most likely to occur, right? Whereas with COVID-2, this whole idea that, we have to constantly get different vaccines, not true, because right now we're trying to deal with this one strain. Maybe years from now, as COVID-2, it's made the jump from animal to human, as it goes back into an animal reservoir, maybe many years, and actually we have 4 coronaviruses that are circulating that cause the common cold, right? So maybe in the future, COVID-2 will go back and become one of these things that are able to circulate, right? So in that case, maybe this will turn to the same case of the flu, where we have vaccines against multiple coronaviruses that are circulating. But there's a whole nother thing that we have to deal with, which to be honest, I shouldn't have gone down this road, right? But it's not the same as the flu. It doesn't mutate in front of your eyes, right? It's this whole guessing game. Okay, I'm gonna stop talking. So last thing I want to say before we take our break, just eukaryotic virulence factors, right? Again, they have them, not as fun, not as diverse, right? But similar themes, right? So, Candida albicans, which is a fungus, uses adhesins to bind. It has enzymes to spread, right? So proteases degrade keratin, which is like this protein in our skin. So it allows it to, once again, break certain things down and allow it to invade farther into tissues that are more sensitive. Cryptococcus trypanosoma, fungus and a protozoan. They have a capsule, once again, protects itself from the immune system. Fungi produce toxins. Mycotoxins are what toxins produced by fungi are called. Some of them are incredibly powerful neurotoxins, by the way. I think we talked about some of these already. And then some protozoans, like malaria, for example, are capable of antigenic variation. And they do it so much better, right? Because they undergo sexual reproduction. So their antigenic variation is part of their nature. They do it not accidentally per se, but because of the fact that they're always breeding, always able to reshuffle their genomes constantly, right? And then some helminths can evade or suppress the immune system. Okay, now is a good time for a break. We've talked about pathogenicity. We're going to move on to epidemiology. I'm going to take a break because I need to get some water, right? But I'll see you once again in a second or like whenever. All right, I'm back. You're back. So let's talk a little bit about epidemiology now. And, we're going to talk, like with everything, just kind of like surface epidemiology. I'm not going to lie. I'm not like an expert on epidemiology. It's rooted in microbiology, just like in everything else. But it's this, it's so much more, right? It's almost like merging mathematicians with epidemiologists. Microbiologists. A lot of it has got to do with statistics and numbers and all this kind of stuff. Important thing to understand is, epidemiology is a study of diseases. It's all about trying to figure out where diseases are, where they're coming from, how often do they pop up, what causes them, how are they transmitted, right? All this stuff is designed to try to control outbreaks before they happen ideally, or in the event that one is going on, how do we slow down or stop this outbreak, right? So epidemiologists are very much involved in disease prevention, outbreak prevention, and then definitely outbreak or disease control in the midst of some sort of outbreak, right? Typically, they're forgotten people. No one cares about them. They're, in fact, super annoying until stuff happens, right? Very important. Epidemiology is, it's data-driven, right? So, epidemiology isn't just this general, this is what I think, this is what I feel. These are just like general things, and numbers have to back epidemiology and the things that we do, right? So all these action plans that we do really need to be rooted in numbers, right? And I think as we talked about at the beginning of this this module, there's so many different ways to interpret numbers, and human populations are so difficult to control that, the job of an epidemiologist is incredibly difficult. But let's start off by talking about some definitions of epidemiology. Many things that, by the way, sound very similar, but are actually different. And these two are, nope, sorry, that's the next slide over. This one is often confused with another, right? Morbidity is the state of being diseased. Sometimes we think morbidity is like death. It's not, right? So morbidity is this, we are like this disease state, and we often relate morbidity in two ways. One is total morbidity, right, which is just the total number. And then the other is the morbidity rate, and that accounts for the population size, right? So the way to tell typically with total morbidity is just some sort of number, right? 10. We have 10 people sick, right? And that will be the total morbidity, whereas morbidity rates are always population size. So it'll be some sort of percent, 5% of the population, or 10 per 100,000 or something like that. Some sort of rate would be, something that accounts for population size would be considered the morbidity rate. And both numbers are important, and they can definitely be skewed however you want according to the point you're trying to make, right? So, because of course, larger populations will have a larger total morbidity. The two aspects of morbidity are two very important numbers. The prevalence is the number of people with a disease at any one time. The incidence is the number of new cases, right? And again, this is this very vague period of time, right? Are we talking a year, a week, a month, right? So let's use HIV as an example. This is from your book, right? 2012, right? That's this last number here. In 2012, we have 1.2 million people with active HIV. This would be, what number would this be? Is this the prevalence or the incidence? This would be the prevalence, right? So the prevalence, I'm just going to put P, is the total number, right? Whereas in 2012, we had 43,000 newly diagnosed cases. This would be the incidence, okay? Now, of course, the incidence factors into the prevalence, right? And then a number of things will factor into our prevalence as well, right? So something like HIV, which is this chronic disease, latent and then chronic, right? We have it for a long time. Because people don't die off fast, our prevalence is going to be very high, right? We don't also cure them very well. So our prevalence will always continue to go up. Because notice here, by the way, the incidence has stayed pretty flat for many, many years, right? So somewhere around here, we've sort of started to get into control. This would be maybe our kind of issue at one point. But now our incidence is relatively stable, right? But because people who have HIV don't die fast, right? The prevalence is still going to go up for a while, right? Whereas something like the flu, which is acute, it'll go much faster. Generally, the prevalence won't continue to increase if we can manage our incidence, right? Good. So then that brings us to when diseases pop up, right? So we'll have things like that's just pop up occasionally, something like tetanus or the plague. These are diseases that we see occasionally. We say they're generally not region specific because they just kind of pop up here and there, right? So yes, believe it or not, we still have incidents of the plague here and there. I think like 3 or 4 cases in the US per year. It's just kind of pops up occasionally. So we will call this a sporadic disease. It's not something we normally see. It's definitely not something you planned for, but it does happen, right? An endemic disease is something that's constantly present within a population, right? And then, so typically diseases are endemic for specific regions, right? So, like the cold, we could consider that endemic. At any one time, someone's going to have the cold. Anywhere in the world, someone's going to have the cold, right? So endemic, the cold would be endemic. The flu would be endemic for a lot of regions. The levels that are, the endemic levels are different probably for different places or regions, right? But they're going to be like that. Maybe a bit more specific, malaria is endemic to subtropical regions, right? in Africa and in Asia, but not in, like, let's say the US, right? So it would be endemic in Africa, right? So, you say, okay, well, here we have X amount of cases of malaria. No one's freaking out because that's like our usual number. Whereas all of a sudden, if you had some cases of malaria in the US, you would freak out a little bit because, like, hey, we don't normally have it here. So in that case, it would be a sporadic disease, right? Same with tuberculosis. It's kind of endemic here in Hawaii, or in Southeast Asia. There are a certain number of tuberculosis cases, right? Whereas maybe I don't even know if this is true, but maybe in Europe, it's not endemic. I don't know if that's true. So forget what I said. Epidemic diseases are when you have an incidence that's higher than expected, right? Okay. So this would be an endemic disease, this could be an endemic disease, right? Something that we have normally, right? But then all of a sudden it's higher than normal, right? So something like influenza, for example, that's like endemic, right? We have influenza everywhere, right? However, sometimes it spikes. Right, so then in that case, it would be epidemic. So even though we say, oh, we always have the flu, and even in the winter, we say the flu is going to go up, right? We would say then that it's still expected, right? But if it goes higher than normal, we can then say that we are in an epidemic flu year. And we have these occasionally. Sometimes you see that in the news. Some diseases, the incidence is 0, right? So, measles, it should be super low, right? If any. So sometimes we get a few cases, all of a sudden, instantly, that would be considered an epidemic, especially if it's not going away anytime soon, right? So maybe 3 or 4, you're like, let's watch this, right? But measles is so contagious that, typically it'll spread to like 20, 30, 40, right? And so even though that's way lower than the number of influenza, we're not used to seeing these types of numbers. So then that instantly would be called an epidemic. And then if something goes on a global scale, that we call a pandemic, right? So swine flu went pandemic, COVID-19, right? Typically, things that have respiratory origin, we consider them pandemic, or they tend to go pandemic because the spread is much easier and it's much harder to stop, right? So an example then is looking at graphs, right? So this is a graph of emergency department visits for influenza-like illnesses. Some of these are influenza, but they're not necessarily tested as influenza positive, right? So, once again, what do these numbers mean? This would be our endemic level, national baseline. So anything lower than this is okay, right? And then over the years, we have certain incidences, right? So 2006, 2007, that flu season, notice we had this peak. So in this case, we would say that, it went epidemic in 2006, 2007, same with 2008, 2009. Look at this one here, 2007, 2008. That was a big year. That was actually a year that they guessed wrong on the flu vaccine, right? Remember, we just now we talked about how we have to guess the strains, right? Sometimes they get it wrong, and then it's like they put out this flu vaccine, they get it wrong, right? All of a sudden, a bunch of people are getting the flu. Then they start testing for the flu, and they realize, oh shoot, this is wrong. And then they try to backpedal, and see if they can create a flu vaccine that is now matches, right? But there's always a lag, right? So it always takes time to get there. Another way to look at things like this is, looking at our normal threshold. This is our seasonal baseline, right? And this is now looking at deaths due to influenza and pneumonia. And again, we have certain times when, this is the actual mortality. This is our threshold. So these are hypothetical numbers, by the way, right? And anytime our numbers go above the blue line, then that would be considered like an epidemic, right? So in 2007, 2005, we had these excessive deaths. 2008, we just talked about that, right? Excessive deaths. So in this case, clearly something is going on. Epidemiologists come in there. Okay, so what do they do, right? What do epidemiologists do? Well, first and foremost, they always need to find the cause of a disease, right? So, of course, they need to determine that there's an epidemic, right? But in the event that there's some sort of disease going on that they don't know about, they need to find the cause of the disease or the causative agent, right? This etiologic agent, right? Etiology is the study of causes of disease, right? So the etiologic agent or the causative agent, that would be that's one of the first things they want to figure out. And sometimes it's difficult, right? Because sometimes signs and symptoms are non-specific. So COVID-19, for example, a good example, it came into the hospital in the Wuhan province as abnormal pneumonia. And, they track these things, right? Every year you're going to have some sort of weird abnormal pneumonia because you don't quite get this the cause right away. But there was an excessive number of cases of abnormal pneumonia. And at some point they realized, oh shoot, you know what? I think we have a problem, right? So then you want to go and try to find the causative agent, the etiologic agent, right? And back in the day when during the SARS-1 outbreak, took a lot of time. In this case, I think it was a matter of maybe a couple months where they were able to, maybe even a month. Definitely in terms of sequencing, it happened real fast once someone realized what to look for, right? And remember Koch's postulates, right? This idea that we use these set of rules to figure out what the etiologic agent is, right? So it's Koch's postulates then that help us through experimental measures determine what the cause of the disease is. Okay, so who deals with this kind of stuff, right? And so we have these public health organizations. In the US, it's the CDC, Center for Disease Control and Prevention. I always find it funny, by the way, that CDC is called the Center for Disease Control and Prevention, right? What happened to this P? How come we don't call it the CDCP? But anyways, and so they have number of functions, right? And one of them is to regulate this body that's part of the CDC. And I don't remember what they're called, but it's in your book. You can read it, right? And what they do is they have these notifiable or reportable diseases, right? So when you go to your doctor and they diagnose you with a certain disease like Campylobacter, for example, or chlamydia, these are notifiable diseases, right? So at the end of every week, they say we have this many cases of chlamydia or Campylobacter or whatnots, right? And then they send that report off to the CDC, and what the CDC does then is they compile this report, this morbidity and mortality weekly report that then goes back out to everyone else, all these other state-level public health organizations for then tracking, right? And so then if you're looking at, the normal numbers, like, oh, we have X amount of Campylobacter, right? Then we're okay, right? But all of a sudden it's like, holy crow, we're so much higher than normal. It's like, okay, this is kind of a problem. And then from state to state, you're going to often have specific notifiable, reportable diseases. So something like, Lyme disease, I think that's reportable. In one state might be something that's constantly watched for, whereas in other states, like they don't even have it, so they don't bother with it, right? So again, some of these state-specific ones can vary. Okay, so let's talk a little bit about history, right? I like history, right? So how does one track this kind of stuff? What do epidemiologists do, right? And then we often have these classic epidemiologists that have kind of pioneered the way for how we do things now, right? So what they used to do is still done to many levels today, right? Or on many levels today, right? Just often with maybe advanced technology, right? But the general framework is still there, right? And it's all started mostly by physicians who adopted this germ theory. And remember way back in our very first module, we talked about this idea that germs cause disease instead of miasma, right? This bad air. And so once physicians realize that there's something that causes these diseases, right? They were then able to try to figure out systematic approaches to to prevent disease, right? So once again, data-driven, very systematic. Everything is organized, right? So we're going to start off with probably one of our most famous epidemiologists, often called the father of epidemiology, John Snow, right? This John Snow, by the way, he knows something, right? And so what he's famous for is deducing the source of transmission during a cholera epidemic in London in 1854, right? And so these were actually a series of epidemics, right? But very famously, what he did was he started mapping where the people who got sick lived, right? So he just started mapping that, right? And he started to notice there's this cluster of people around this one well, the Broad Street pump, right? Where they got their water from, right? And then, so then he recommended, based on that, why don't we remove the handle from this well so you can't get water from there anymore? And then they did that, and then the number of cholera, the incidence of cholera greatly reduced suddenly, right? Simply because people weren't getting water from the well anymore. And then when they did further investigation, it turns out there was like a leak from sewage into that well, right? So that was a sewage-filled well. my favorite part of this story, by the way, is not everyone around this well got sick, right? They had a bunch of people that lived around there, right? And then there are a whole bunch of people that didn't get sick. Turns out that these people who didn't get sick, they were drinking beer instead of water for their sustenance, right? So, even though they lived around that well, they weren't touching the water. They were drinking beer instead, which highlights the universal theme of life. Drinking beer saves lives. So this then leads to a couple more definitions from this, right? Where does disease come from, right? So we have this, or where does it spread from, right? What's the source? So a common source spread is one where there's a single source for all infected individuals, right? So the source is this one place that the disease is continuing to spring up, right? And then we can have a point source where it just exists for a short period of time. So let's say we had this sewage leak, and then the sewage leak got fixed, and then all of a sudden now it's not going to be a source for spread anymore. That would be a point source, right? A continuous common source would be one that exists for an extended period of time. So this continual leak of sewage into this well that no one ever fixes, right? So this thing is continuing, be this continual source of this pathogen, right? And an intermittent common source would exist for a short period of time, stop, and begin again. So something like this then would happen for, let's say there's heavy rains, there's a lot of runoff that pushes sewage into this well. When the rains go away, and then eventually the sewage gets cleared up, now our well is no longer contaminated, and then the next heavy rains brings some more sewage, etc., 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 right? So that's when it's one single source. A propagated spread is no single source. In fact, it's many sources. And this is usually via person-to-person contact, right? Because the source then will start to move around. And then so you can graph this actually, right? So if you have, let's say, a point source spread, right? Typically what you'll see is this one large incidence and then this tapering off, right? Because we have this one point source, typically it's then found, and then your incidence typically doesn't go, or it'll stay the same, right? Regardless, things will be constant as you're continually interacting with this point source spread. Whereas with propagated spread, you'll have these continual peaks and something like that, right? As the people who are propagating the spread are moving from source to source, right? Are spreading disease just through interaction. So sometimes they're going to be hanging out with a lot of people, sometimes they're going to hang out with less people, etc., etc., etc. So you can actually graph this and start making hypotheses as to what the spread or potential source is, right? Once again, data driven, right? Florence Nightingale was a nurse in the Crimean War, which was like somewhere in Europe. Battle against Russia, I think it was, some sort of religious battle. They were all like religious back in the day. But it's like persecution of Christians in the something by the something people, right? This war that we never ever hear about, but we all, I think, heard about Florence Nightingale, right? She's like this prototypical nurse, right? The nurse figure that cares for everybody, right? And so her claim to fame is this just meticulous record keeping, right? So as a nurse, for her, it wasn't sufficient just to take care of the soldiers during the war, right? She took records of illnesses, of deaths, how they died, how they got sick, and then I already forgot the thing, but it's in your book, right? So I think this wedge here shows number of deaths from injury. This one was number of deaths from something, and this one was number of deaths from not war related, right? And basically her numbers showed that the largest cause of death really was due to poor sanitation. So, most of the people that died in this war didn't die from like bullet wounds. It's because they were like hanging out next to somebody who was like making doo-doo right next to them, right? And they're just kind of shoveling it over, thinking that they covered it up, right? And then they're like lying in it, right? So they're getting sick because of these really unsanitary conditions that they were in. Right? And so when someone looked at this data and they realized, oh my goodness, I think this is way too much death. So we need to start, maybe everyone makes doo-doo over there, not in the place where you go to sleep and all that kind of stuff. Let's really start thinking about, the conditions that we're in, right? So that's kind of what her claim to fame was, right? Again, this idea of generating data, right? Keeping records, very meticulous, and then using this data to come to some sort of conclusion. We talked about this person before, Joseph Lister. He was a surgeon, right? He was the fellow that said that it's surgeons that introduce fatal infections, right? Because they are transmitting infection from patient to patient, right? And remember, the prevailing theory at the time was that surgeons, we are clean people. We are not responsible for this, right? But he did provide evidence through controlled experiments, not controlled experiments, but through looking at data, looking at surgeons that washed properly, that used phenol to sterilize their tools versus surgeons that did not do that, right? And then he showed very clearly that the infection rate was like ridiculously lower, like so much so that, I mean, anyone who was saying that Joseph Lister was wrong, and by the way, people believed he was wrong before he did this work, right? Anyone who said that he was wrong was really just being stubborn. That's really all that was, right? So let's talk about the types of studies then. It is a science, so it's all about hypothesis testing, right? We have a hypothesis, right? So, the hypothesis for John Snow was that, well, I believe that it's this pump that is causing the disease, right? Or that's the source of the disease. Florence Nightingale says that let's clean up our sanitary conditions and then our soldiers won't die needlessly, right? Lister said that let's clean our tools, let's wash our hands, and then maybe our patients won't die from these infections, which we feel like are magically caused by something else, right? Two general types of studies, observational and experimental, right? Observational studies are all about gathering data, measuring data. We don't really our subjects or our patients are not manipulated, generally easy to perform, right? No ethical issues, because you're not going to say, well, let's infect these people and let's not infect those people, or let's withhold drugs from these people, right? But it can only identify correlation, not causation, right? So just because two things appear to be related doesn't mean that that there's a causation, right? So John Snow's experiment was observational, right? It wasn't through any manipulation. It's just this correlation. Same with Florence Nightingale, right? They took these data. They say, hey, look, there's this correlation between this pump and the people getting sick of cholera, or these unsanitary conditions and then the people dying from it, right? And so then they took the measure of dealing with it, and then it turns out that in this case there was causation, right? Experimental studies are things in a controlled setting. We have a control group, our non-control group. So Lister's experiment was more of an experimental, not in the sense that he says, okay, all you folks go cut these people open and don't wash your hands, and you folks do this. But he did take the data of people that did it and the people that didn't, the control group and the non-control group, right? And then use that data. In this case, there was this clear link between cause and effect. And again, it's somewhat, sometimes the lines are a little bit blurred, right? But generally, experimental data, much more useful, right? But it can be difficult to design an experiment that is ethical, right? That will show us clear cause and effect, right? A very good example of this was that hydroxychloroquine, let's call it a debacle during COVID-19, right? Early on, people had these anecdotal observations, right? Hey, I gave my patient hydroxychloroquine, they got better, right? Hydroxychloroquine is an anti-inflammatory, right? So it has helped with the response. They got better, right? Of course, they gave it to like 12 patients, and these 12 patients are getting so many other things that really, this is a true correlation, not causation, right? So when they did some experimental studies, when they had test groups of patients, and they said, okay, we're going to give these groups hydroxychloroquine, and then these patients, we're going to give them a placebo, they can clearly see all of a sudden that there was really no difference in the recovery rate of people who were getting the hydroxychloroquine versus not, right? And in fact, it appeared that people who were getting hydroxychloroquine were doing worse than the people who are not getting hydroxychloroquine, right? So this is one of those cases that they set up this experiment. Once they started the experiment, they realized, oh shoot, this is actually not good. We can't finish this experiment. So they stopped these experiments, and it immediately took people off hydroxychloroquine, right? So again, this idea, experimental study is much more powerful, right? But ethical issues can arise, especially if someone decided we're going to continue with this this experiment because we want to finish it out, right? In this case, it would have been a bad thing to do. Okay, so that's our experimental our how epidemiology works, right? Let's talk a little bit about disease transmission, because understanding transmission is very important to preventing spread. We want to know cause, of course, right? The etiologic agent, but we also need to know how disease is spread, because once we're in the midst of a pandemic, actually anything, any epidemic, right? Even a sporadic disease, if you know what transmission or how diseases get transmitted, we know how to respond appropriately, right? Generally, we already talked about communicable versus contagious versus non-communicable. And then not all diseases transmit the same, right? So communicable diseases have different methods of transmission, all right? So, we'll talk a little bit about the source, right? The reservoir of infection is a continual source of a pathogen, right? So the important thing here is that the pathogen is able to grow and replicate. Reservoirs can be non-living, like soil, water, food, right? Or it can be living, like humans and animals, right? Sometimes this can be confused with our point source, right? So it really depends on what manner are we talking about, right? In which way are we talking about this reservoir of infection? Now, carriers are capable of transmitting the pathogen without displaying symptoms. So many reservoirs, like a human reservoir, for example, will be a carrier, right? Because they are capable, because they are a source of the pathogen, and then they'll transmit, but they themselves don't display symptoms, right? And there are many diseases in history that have been like this. Passive carriers are not infected, right? So they'll have like the pathogen on the outside. Active carriers have infection, but the disease is either mildly symptomatic or asymptomatic, right? And so then they can carry the disease, right? An example, really good example of this was polio, right? So polio was this, we are, I think, all way too young to even know about polio, right? But polio was this disease that it was just a scourge for kids, right? And I think in Europe at some point, you kind of assume your kid would get polio. And then out of any kid that gets polio, I think something like 10% of them ended up paralyzed, or maybe even higher than that, right? So, but so many people were asymptomatic, right? So this really neat case then of we have all these carriers. You don't know who's carrying it, right? But then they can spread the disease, right? So that was a very important thing until we were able to finally eradicate it. Animal reservoirs are called zoonotic diseases, right? So I feel like we talked about this before, but we'll just mention again really quickly. Zoonotic diseases are when the reservoirs are animals, right? Animals themselves may be symptomatic or asymptomatic, right? So for example, influenza at its root is a zoonotic disease because it lives in birds. Almost all birds have some sort of influenza virus, right? But they live there asymptomatically, right? But they can also be symptomatic, right? So you can get rabies, for example, from a dog, right? Rabid dogs are symptomatic, and then when they bite you, you're going to get rabies, right? And then parasites that have complex life cycles, that have multiple hosts, will often use each host differently, right? So the definitive, sometimes primary host, the parasite undergoes sexual reproduction. The intermediate or secondary host, parasite reproduces asexually. Okay, so these are two very different uses for the host, right? Very complex life cycles, right? As it has that interplay. And again, in this case, parasites that have multiple hosts have to go from host to host. Okay, then, so let's talk about how diseases are transmitted, right? 3 general methods. First is contact transmission, and contact transmission involves contact, right? Turns out there's different types of contact, right? You can have direct contact, which is person-to-person touching, that's direct contact. Generally, we talk about horizontal contact transmission. That's kind of like people to people, right? But you can also have vertical contact, which is mother to child, right? Generational almost, right? So sometimes there are certain infections or diseases that are very significant as the child leaves and passes through the birth canal. Indirect contact is person-to-person contact via some sort of inanimate object, and this is called a fomite. Right? So a fomite is any inanimate object, whether it's like a pen or a tissue or a table or something that allows for contact to happen. Now, Important thing with a fomite is that fomites do not allow growth of the pathogen. It just kind of hangs out there until at some point it dies, right? So it's not a reservoir, which allows pathogens to grow. It's just a place where the pathogen hangs out. And then finally, droplet transmission is transmission via mucus droplets. So this is important. And that's because the pathogen lives in these mucus droplets, right? And then they're of some certain size, so they carry some certain weight, right? Generally, by definition, we call droplet transmission to be about 1 meter, which is 3 feet. And that's because when you cough, sneeze, or talk, the mucus droplets are heavy and they'll fly and then fall down at a distance of about 3 feet, right? And we'll talk later, well, and then this continuum, we'll talk later on the next slide actually about this kind of distinction between what is droplet and what is airborne, right? Because it does pass through the air, but it does at some point drop, right? Vehicle transmission is transmission by some sort of, quote unquote vehicle. And that's basically water, food, air, right? The pathogen has to ride on one of these things to get to its, from host to host, right? So waterborne, you got to drink it. Foodborne, you got to eat it. Airborne, you got to breathe it, right? So, and the pathogen happens to be there, and then you're able to take it in. I do want to talk a little bit, pretty straightforward, but I do want to talk a little bit about airborne transmission, right? With airborne transmission, we're talking about great distances, right? So sometimes it's hard to differentiate between droplet transmission and airborne transmission, right? Because it's not like this line where it's like, okay, 3 feet, bang, done, right? That's droplet, and anything after that is airborne, because there is this continuum, Some pathogens, they need mucus, but they can survive in smaller drops, so they can actually go a little farther, right? Some, they don't, right? So, we have our extremes where they need thick drops of mucus. They will be kind of at the short end of the continuum. Some, they don't need it at all. And then just simply the matter of speaking, like measles is airborne. You just speak and then it just goes. Great distances, right? Others have this continuum. So I think for SARS-CoV-2, we've adopted this 6-foot thing because it seems to spread just a little bit farther than normal droplets, which is why at one point they thought maybe it was airborne transmission, right? Which is crazy, right? It's always more dangerous when it's airborne transmission, but the spread didn't seem to, the speed of the spread didn't seem to indicate airborne transmission. Airborne transmission spreads so much faster, right? So the hypothesis now is that it's able to survive in smaller droplets, right? So that's why we're kind of within that 6-foot rule. The third type of transmission is vector transmission. And vector is through some sort of animal carrier. Typically, we're talking about arthropods, right? Insects, arachnids, right? Which are like mites, crustaceans, myriapods, which are like millipedes and centipedes. 2 types of transmission: mechanical vector, which is basically the animal carries it without being infected. So it just like walks on some doo-doo, picks up some bacteria on his legs, and it lands on your food, and then now you're eating this stuff, right? Or biological vector transmission, which is transmission that occurs as part of the life cycle, right? So this is very specific very specific pathogen transmission, whereas mechanical is just like this non-specific, just kind of happened. Okay, let's play a little game. Let's look at some pictures and identify the type of transmission, right? So I'm gonna, for each one of these, let's give it a couple seconds. What type of transmission are we trying to depict here? And of course, you can see many things, right? But here we're trying to show, this is an example of direct contact transmission, right? We're having this really fun party, eating food, and then we're like, oh, ha, this is the most hilarious joke I ever heard. And then, through contact, we can then transmit, so if she's sick and she got some droplets on her hands, now she's sick, or she's going to get those droplets, and then she's going to go and scratch her nose later, and then boom, now she's got the flu. What are we trying to show here? Like you said, foodborne transmission, that is correct. This would be an example of vehicle foodborne transmission, right? So, we all know, wash your hands, don't cut raw meat on here, right? Many, many examples of bad sanitary practices. These are always my favorite pictures. What are we trying to show here? This is contact transmission, right? Droplet contact transmission, right? Because the stuff has to be in these mucus droplets. By the way, no one sneezes like this. Don't think this is what a sneeze looks like, right? They give these people like water or some sort of thick liquid and they say, okay, you sneeze now. And then they sneeze and makes this really disgusting spray. just to kind of measure how far these things go. But no one sneezes like this, I promise you. If they do, don't hang out with them. Money, money, money. What are we trying to show here? Here, we're trying to show indirect contact transmission. Money can act as this intermediate, this inanimate object, which we call fomites, right? So sometimes we actually call this fomite transmission. How about here? What are we showing? Doesn't this look like a Lord of the Rings scene or something? Waterborne transmission, vehicle waterborne transmission, right? Here. This is rat lungworm, I think. Yeah, this would be biological vector transmission. Transmission as part of the life cycle of a pathogen as it's going from host to host, primary and secondary, definitive and intermediate, right? How about here? What are we trying to show? In this case, this would be mechanical vector transmission, right? Hey, I got a fun thing for you. One time I was goofing around in the lab, right? And then, so we use fire in the lab, and then there's this fly. It just flew right through my fire. I don't know what the heck it was thinking. And along the way, it burned its wings off. I'm like, oh, that's so sad. Did I feel sad? I don't know. And so I was gonna like squish it and put it out of his misery, but then I thought, you know what would be really fun? Let's take this thing and put it on an agar plate. So I stuck it on an agar plate. It's what we use to grow bacteria. And then I let this thing crawl around for a little bit before I put it out of its misery. And this is what came off it, right? So again, this idea that it's carrying some sort of bacteria. Now, that doesn't mean these are pathogenic, by the way. I have no idea what bacteria these are. I never did the follow up on this, right? And to be honest, if you stick your finger and rub it around this plate, lots of bacteria are going to come off of that, right? But this generally, like let's say this thing was some sort of intestinal pathogen, right? That it picked up while it was goofing around and doo-doo somewhere else, right? These are the types of things then that can be passed on. It didn't intend to be passed on to you. It wasn't part of its life cycle. It just got there accidentally, right? Mechanical vector transmission. Okay, so with that said, now we are sick. Now what do we do? What's our response, right? So the response is always going to be very, again, specific to the pathogen, right? So, if you have a known exposure, even suspected exposure to a contagious pathogen. Remember, contagious, what does that mean? Contagious refers to what? What? Right? Easily transmissible, highly transmissible, right? So if something is very contagious, you're going to have to quarantine, right? And then the level of quarantine is really going to be disease dependent. Even the mechanism, the facilities you put them in, right? So if you have like this world killer, if you have ever watched the movie Outbreak, right, with airborne Ebola. Ebola is an extreme killer. 50% of you die if you get Ebola. If you don't treat it, 95% of people die, right? And then something that's airborne where it can spread, right? You need to be put in a special facility because that stuff is going to spread, is going to kill people, right? But for some things, respiratory issues, for example, you look at something like this and you think to yourself, hey, man, how can you stick a bunch of sick people here, right? But remember, respiratory droplets, 3 feet, 6 feet, right? If you like alternate beds or if these beds are 6 feet apart, technically we're all okay. And this always reminds me of the story. I was talking to this nurse once and she was complaining about, this was when MRSA was a big deal. Remember when MRSA was a big deal? So she was complaining about, what our control mechanism is for someone who tests positive for MRSA? We just close the curtain. And she was like trying to say that with this whole disdain, like MRSA is going to jump from this person and like jump down her throat, right? But remember, MRSA is contact dependent, right? It doesn't transfer through droplets. It doesn't do anything. It's through contact, right? So you know what? A curtain is more than enough to prevent MRSA transmission, right? So you simply quarantine them. You don't even need a curtain and then you're good, right? It's same with Ebola, right? Ebola is contact through fluids, right? So if you put this person in a room and then you don't touch their fluids, you will be fine being in the same room with them. I promise you that. Not my recommendation, by the way. Got to be careful if you ever find yourself in that situation. But, you have to understand, right? Whereas things that are droplet, you got to think about how that's going to get to you, or especially airborne. That's extremely important, right? All right, so the last thing I want to talk about is this idea of global public health, right? Public health is such an important thing. We never think about it, though. until it happens, right? I always like to think of these as like insurance, right? You know, you pay for insurance every year. Every time you pay for it, you're like, oh, I gotta pay for my insurance. Gosh darn, Geico's taking my money for what, right? And then you get into an accident, and then Geico gives you money back and helps you fix your car or stuff like that. And you're like, okay, I can see the value of insurance, right? And so it's the same idea with public health. You never think about it until you know it, right? Especially global public health, right? Because, you know, especially in this day and age, pathogens, they don't care about boundaries, right? So just because you know, some outbreak started in China or in Mexico or in the US, right? They don't care. They're not going to be like, hey, I'm a Chinese pathogen, I'm going to stay in China. That's not how it works, right? They're just going to go everywhere. So having a coordinated global public health plan is so vitally important. We all know about the World Health Organization, right? It's our largest public health. And then we have multiple smaller organizations. We have the CDC for each country. And then within each country, of course, we have multiple public health organizations, all with this idea of trying to coordinate and track diseases, monitor responses or coordinate responses, and then deal with these things, right? The problem, of course, now is sometimes public health issues are politicized, right? Which is always the wrong thing to do, right? Because, you know, public health should never be politicized, right? It is our health after all. So, you know, trying to politicize things and then making decisions based on politics will lead to great number of death and suffering, right? Some of the things that the World Health Organization has done is eradicate smallpox, right? So it started this program from the 1960s. Smallpox, by the way, you know, we think of this as this biblical disease, right? But it used to be a huge issue. People died from it all the time, right? But, you know, so I think in 1980, they said no more smallpox. Polio, no more polio, right? Like this kind of stuff is almost unheard of nowadays, right? They have a lot of things in these smaller countries. We're dealing with malaria, with TB. HIV was a big issue in the 80s, right? So, you know, what was this response to HIV? And then non-infectious diseases as well, stuff like diabetes, right? So it's the health organization, not infectious diseases, right? But their number one role is to coordinate this international response during global emergencies. So it's all about feeding information. You report to them. They report numbers back. They make the recommendations. You do what's important for your country, and then try to have this thing to stop, right? Because if everyone in the world is participating in public health, but 3 countries are not, well, this whatever disease is going on is not going away, right? So then that brings me to the last couple of things, this idea of emerging infectious diseases and re-emerging infectious diseases, right? So, you know, there's this list of what they call emerging infectious diseases. I forget how long it is, right? But basically, they're extremely dangerous because they are either new or extremely deadly, right? With no therapeutics. So Ebola, for example, is on this emerging infectious disease list, right? Stuff that's always watched because it's still relatively new. We don't have any ways to treat it really, right? And then basically it's defined as this increase in prevalence in the previous 20 years, right? So, you know, there was this initial outbreak here that happened, right, when we first learned of Ebola, and then really nothing happened. Although I think maybe it's in your book, I read somewhere that, you know, chimpanzees and gorillas were dying of Ebola, right? But that was their version. And then somewhere along the way, the jump to humans came, right? And then we had that big one in 2014 where a bunch of people in Africa got it, right? And just, you know, because of their cultural practices, it really spread to a lot more death than should have happened, right? And so these things are very important to control because the population is much more susceptible to diseases we've never really seen before, right? So when we don't have any natural immunity to these things, very important, right? So we think about, oh, you know, the common cold. Well, one time, the common cold probably killed a bunch of people, right? But over the years, as our human population has adapted to it, you know, it's now kind of this, it doesn't even cause us problems anymore, right? So these diseases are still new to the population, still quite significant. Kind of related to that are the re-emerging infectious diseases, right? Those that it appeared to be in decline have now come back, right? So, you know, H1N1, the swine flu, you know, that was first seen in 1918, and actually it kind of disappeared for a long time, and then it came back in 2009, and that freaked a lot of people out because the 1918 was this super deadly flu virus, right? So it killed millions of people worldwide, right? So they thought, oh my goodness, here's H1N1, it's back with a vengeance, right? Luckily, was not The mortality was much, much lower, right? So it wasn't so bad, but it could have been bad. That's what people freaked out about big time, right? And then, so there are a lot of reasons why this kind of stuff can come back. So drug resistance, climate change, whooping cough, I think at one point was a re-emerging infectious disease because we have a vaccine for it, right? But then over time, as it mutated, there were strains now that were resistant to the vaccine, right? So there's this constant warfare. Sometimes climate change, as we cut things down, certain viruses, they really don't have their vector that's required to carry them, right? But then all of a sudden you cut things down, in come these rodents. Now the rodents can carry disease, right? So what used to not be a problem is now a problem, right? So all these things kind of factor into it. Okay, so that's, I know, long module, right? But really cool stuff, I hope, right? Stuff that's extremely relevant to you, right? In this population. You'll see it in the news all the time. Next up is going to be Module 10. In Module 10, we start talking about us, right? Our immune system, right? Module 10, 11, 12, we start talking about our different arms of the immune system and then some issues with the immune system. Okay, so luck on the quiz. I'll see you soon in Module 10.